Hello, and thanks for joining us today for this iRepertoire webinar. My name is Matias, and I will be your moderator for today. In this webinar, Mariska Haak, Senior Director of Analytical Development at InnateBio, will present about evaluating gene expression and TCR repertoire dynamics in gamma delta T cell therapies. Thank you again for answering the polling questions. And now, without further delay, I will turn the webinar to Mariska.
Hello, thank you, Matthias, for the kind introduction. And first of all, I also want to thank iRepertoire for giving me this opportunity to share our data on this platform. So today I'm going to talk about evaluating gene expression and TCR dynamics in gamma delta T cell therapies. Before I move on, I'm just going to give you initial an idea of what gamma delta T cells are. Then I'm going to talk about a little bit about a program initially. Then I'm going to tell you about a study design and how we utilize iRepertoire's technology for our analysis. And then I'm going to share you the data that we collected in the end. Moving forward, just a disclaimer, as always. So what are gamma delta T cells? Gamma delta T cells, I see them actually as a special task force within our immune system. So they have an innate capability, but also have an adaptive capability. They are MHC independent and can actually have the same persistence as alpha beta T cells, but without the toxicities. They're able to recognize unhealthy tissue, such as tumor tissues, from healthy tissues independently, and have the same killing capabilities of NK cells, but with better durability. So how do they do that? Well, gamma delta T cells actually recognize NKG2D ligands, which are upregulated when a cell is stressed. So once these signals are upregulated, gamma delta T cells will react and kill independently these cells. In this way, they will be able to produce peripherin and Gram-Sm B, and this can even be further enhanced by monoclonals if needed. So again, why gamma delta T cells? Why are we investigating gamma delta T cells in cancer, for example? Well, they actually key for better survival. And as if you look at this slide, you can actually see in the right that gamma delta T cells, if they are elevated in patients, they actually have a better overall prognosis. If you look at in the middle of this slide, you can see for a liquid tumor that actually high levels of gamma delta T cell result in better survival and longer survival. And the same can be said here in this example for colorectal cancer, where you actually see an improved patient disease free progression, while there are high gamma delta T cell levels. Well, there are even lower gamma delta T cell levels. This actually, the progression-free survival actually decreases, and this is even worse when there's low levels of interferon gamma. So this tells to us that gamma delta T cells are actually key to survival for cancer. So talking a little bit about our autologous DeltEx DRI program, before I go into nitty-gritty details, I just want to tell you where DRI stands for. That is a drug-resistant immunotherapy. So we move forward in a clinic for an autologous DeltEx DRI program, and we chose our cancer to be GBM. So glioblastoma is actually a very difficult cancer to treat, and there's very limited treatment availabilities out there. So why did we choose it? Well, we chose it because the brain offers actually a separate compartment to allow to directly deliver the cells via the catheter into the site. So this will actually reduce any trafficking variabilities that might be introduced by other infusion technologies. So we could use the catheter to directly get our gamma delta T cells at the site. Not only that, it moved us forward to our allogeneic cell therapy in solid tumors and simplifying dealing around with graft versus host disease and persistence in the delivered cells. Another advantage is that the brain is considered one of the immune privileged organ centers, which again allowed us to evaluate this more clearly. And in addition, the neuro-oncology standard of care is Smelldar, and we didn't need to introduce a new separate lymphodepleting protocol. So synergizing all these interventions together, we were able to actually make the tumor more fragile and be able to highlight our gamma delta T cells for the tumor and in the hope to eliminate the tumor further. So how do we highlight these cells? Well, we came up with a genetically modified gamma delta T cell. So while you use chemotherapy, actually stress ligands are upregulated in the tumor cells. So while you would actually treat tumor cells with TMC, you would actually see increased expression of NKG2D ligands, which I show here at the right. And the same can be said in gliostem-like cells. So how did we generate a drug resistance or chemotherapy resistance gamma delta T cell? Well, we actually integrated MGMT into the genome of our gamma delta T cells, which is a DNA repair enzyme. So in this way, our gamma delta T cells would be protected from the chemotherapy. So while highlighting the tumor cells even further with TMC, we were actually able to protect our gamma delta T cells so that they were actually as the strongest and be able to eliminate the cancer cell further. A little bit of information about our phase 1 study, IMB200. So we had 3 treatment arms that we dosed into patients. We started off with 1 dose and escalated further to 3 doses and further escalated in a total of 6 doses. We combined them all with 6 cycles of TMC. But what I want to highlight in this slide is actually we performed a surgical resection to remove the tumor initially from the patient so that we could actually generate an autologous product and collect an apheresis products afterwards when the immune system recovered. So afterwards, the patient would go into the daily radiation before we eventually be able to infuse the patient and treat the patient with chemotherapy. So how do we manufacture our gamma delta T cells? So briefly, an overview of our process. So we obtain a peripheral blood via leukopak. This leukopak is further prepared as the starting material and gradally separated so that we could get mononuclear cells. So this is the vital step, starting point of our material. We activate, transduce, expand, and cell separate if needed. And then in the end, we will harvest the final gamma delta T cells when they're ready. In the end, we will cryopreserve them before they're finally infused into patients. What I want to highlight here is that gamma delta T cells, because they're only 1 to 5% in the blood, the processing time and expansion time just takes a little bit longer. And so our processing time is significantly different compared to other cell therapy products such as alpha beta T cells. So our process time is about 9 to 14 days. So we wanted to know if there was actually an impact of this longer incubation time and would have an impact on our process, that the process would actually have an impact on our final products. So we started a collaboration with iRepertoire to be able to look at the TCR sequencing to be able to learn more about gamma delta T cells, but also about what the starting product would be and the harvest the final product that will be infused into our patients. So we set up this study design where we started off with 5 products per group, but we also next to the disease patients, so autologous GBM products, we also looked at healthy donor material, which we used for process and validation studies. And then we compared the starting product towards the harvest product, the final product. So that gave us a total of 20 samples to analyze. So why did we do that? What was the strategy behind this? Well, we had some questions about, like, is there a difference between autologous versus allogeneic? So how important is that donor selection? We wanted to know if there was any difference between manufactured on our manufactured products when we would start with disease state patient versus healthy donors, because we also wanted to know if there was a difference between our process developments and validation studies compared to our final product. We also wanted to know if the profile and the cellular characteristics of the donor were persistent throughout manufacturing. So was it donor driven? Was it manufacturing driven? So next, we also wanted to look into the process characterization and wanted to be able to additionally predict any expansion and activation markers. And we also wanted, as what I said before, particularly the longer manufacturing of the gamma delta T cells process, if that actually had an impact on the final cell therapy product. And we also wanted to further characterize our product and see if we could find and identify potency effector and target capabilities of our cell therapy products. This is as well, if you are in the cell therapy field, you know this is discussed, and it's kind of an important topic in cell therapy, potency, how to do that, how to characterize mechanism and action, because a killing assay is just not sufficient. So, and then we also wanted to look into if actually how producible is our process in the end and robust among different donors. So we utilize iRepertoire technology to characterize our product further. So what is the technology that iRepertoire uses? So they use a dimer-avoided multiplex PCR, where they combine their RepSeq together. In this case, we combine together the RepSeq together with the ImmunoSight technology of them. So the DAM PCR actually removes unwanted product formation while increasing the target signal, but it also allows you to do direct RNA quantification with human eyes, so unique molecular identifiers. But with that, it also allows you to do immunophenotyping panel next to the already multiplexing power that you have for all TCR chains coupled to the NGS. So in our case, we were mainly interested in the T cell repertoire, but you can even combine this together with the B cell repertoire and further multiplex, increase your multiplexing power. So in our study, we looked at the alpha, beta, gamma, and delta VDJ chains, and we looked at immune cell markers up to 150 genes. These genes these genes were targeted to T cell markers, innate markers, cytokine markers, even B cell markers, but also cytotoxicity molecules, activation markers, checkpoint markers. So we could identify additional genes that might be of interest next to the TCR repertoire of our products. So while we're looking at the workflow of the iRepertoire, I want you to also focus on here that we have our starting products and we have our manufactured product. So I'm going to refer to everything to ease out the graphs and the data analysis further by A and B. So the starting material will be A. We will refer to A throughout this presentation, and the manufactured product will be referred to B throughout this presentation. So the RNA of our cells were actually further analyzed, further prepared for an automatic library preparation by iRepertoire. And what I want to focus here as well is that Next, we didn't do single cell sequencing, we did bulk cell sequencing. Why? It would allow us to do actually analyze more cells than when we would do a single cell sequencing. And the other pro about this as well, it also significantly reduces the cost because you're not doing single cell preparation. This final library was eventually analyzed by NGS or sequenced and further analyzed if using the trimming and the further data analysis programs that iRepertoire supports. So all data is actually represented on their IREP, and that allows you to look exclusively at the TCR sequencing analysis. And that eventually also gives you these nice QR plots, which are actually tree maps. And I want to explain a little bit more about that because each individual clone here is represented by its color. And so the more different colors and normal boxes there are, the more diverse your sample is and the population. And the bigger that box is the more of those clones are actually observed. So it gives you a nice idea, nice tree map, a nice idea, overall representation of your TCR repertoire. So coming for our first data that I'm sharing with you is we first wanted to look into the diversity of our starting product. So we wanted to understand if a diseased patient, a diseased starting material versus a healthy starting material would actually have a different diversity in T cell repertoire. And as you can see is that even though that might individually be different, if you look at the different chains, alpha, beta, delta, and gamma, and we look at in here in yellow, the diseased group, and here the healthy donor group, is that those products, the starting products materials, the diversities actually are not that much different. So the diversity is independent of the starting material, which it's still probably still donor dependent, but it doesn't matter if we would start with DBM, a material obtained from a GBM patient versus a healthy donor patient. So while we analyzed the TCR repertoire, the initial analysis, the overall analysis I'm going to show you here is the overall reads that we were able to get with this technology. And if we look at the starting product, we can see that mainly it contained alpha and beta reads. And then there was a significantly shift to, in our final product, to decreased alpha and beta reads, whereas the gamma and delta reads were significantly increased. Here further on below are different ways we could look at that. So just looking only at the reads, we saw a significantly increase. Just looking at the percentage reads, which are presented here as well, you can see the significant reads where in blue the significance is provided, being 0.05 or lower, but also the unique CDR3 clones levels, the gamma delta is significantly increased. So we had a significant increase of gamma deltas. Further on, you could also look at overall at your data, and we also were able to look at the variable gene of, so look specifically at the VDJ, your clones that were presented. And what we were able to, which the data actually showed us, is that we actually had a preferential expansion of V gamma 9 and V delta 2 clones, which you can actually see here in the right bottom of this graph, where if we just look at the individual V clones that were represented by delta and gamma, that there was a significantly increase towards V gamma 9 and V delta 2 clones. If we dive further into our analysis, I can show you the individual reads here and go a little bit more in detail. So what you see here on the left is actually a graph of all the alpha, beta, gamma, and delta reads. So in blue and red, you can see the reads, the percentages for alpha and beta. And then in green and in purple, you can see the reads that were represented by gamma and delta. So if you look at the left side of this graph, you will see the individual responses for GBM donor material and manufactured products. And then at the right side, you will see the healthy donor products. So the starting material, you can see generally in all cases, the alpha and the beta reads are dominant. Whereas after manufacturing and the final product, we actually see a significant shift in all individual manufactured products of gamma and delta reads. On the right side of this graph, of this particular graph, you can see I selected here a GBM patient and sample 5, particularly to discuss the details a little bit more further, but you can even look further into your analysis. So what you can see here for sample 5 is a 4-dimensional tree map of that sample. So on the far right side, you can see the legend of that. Where you can see here are the alpha populations and all the different clones that are represented here, as well as the beta, which you can't really see basically because there are just so little gamma and delta reads. There are no, you could, here on the bottom are actually represented to the delta and the gamma populations, which are completely squeezed to the bottom, because this pre-map actually emphasizes the amount of clones and the bigger an amount of reads. So the bigger the box, the more observed clones were. So if we move forward to the manufactured product, you can see that there was a significant shift towards the delta and the gamma for this particular product. And you can see here where the alpha and beta are squeezed up to moved up to the top, whereas the deltas and the gammas are represented here. You can see that there's specific clones here that are actually expressed more compared to other clones. So focusing a little bit more only on the gamma and the delta reads, before I actually go further, I want to explain to you what you're actually seeing here. Is this is a plot of the tree map of the gamma as a starting product and the delta. And then in the middle, you actually see a tracking graph. And in the end, you can see the final tree map of the gamma read here and the delta reads here. So the individual clones that are represented within the population of the cells that we analyzed. So grayed out are actually clones that did not significantly decrease or increase from start to finish. So what you can see here particularly is that we were able to identify here a V gamma delta 1 clone, which we could further track that from start to finish, and could see that in the end it was dominant in our final product. So this particular V gamma 9 clone was dominant in the final product for sample 5, for batch 5. So if we look at the V delta 2, you can see the same, only particularly starting product V delta 2 was already here dominant, and that further was enhanced after manufacturing. And we could see that V delta 9 and V delta 2s in this case were dominant in our final product. If we move forward to a healthy donor product, I also want to share that with you to give you an oversight of what that would look like. Here are sample 9. In this case, we selected sample 9 to share with you. And you can see here the 4-dimensional plot, a tree map of sample 9, the starting products. And you can, in this case, you can actually see the populations of the delta and the gamma clones. But if you move forward towards the manufactured product, you can actually see that they're actually skewed up to here to the far bottom left of that tree map. So again, the gamma delta, the alpha beta reads are actually skewed out, and we see mainly delta and gamma reads in our final product. So if we share again that tracking graph that I showed before, is here you can see again that these two V delta 2 clones further were expanded within our product, and in the end were dominant in our products. And then for V delta 2, where these purple and this pink lilac populations could also be tracked and further were further increased in our final product, which resulted in a V delta 2. So although of course, the starting product is essential, because if those clones are not there, you cannot expand them. But it does tell us overall that for all the data that we analyzed, is that there was a specific a preference for expansion of V gamma 9 and V delta 2 clones that showed actually that our manufacturing process was actually robust and mainly driven to actually produce V delta 9 and V delta 2 clones. After looking at the T cell repertoire, we could also, with the bulk sequencing, look at further also at gene markers. In these case, we looked at immune gene markers, the 150 genes that I spoke about earlier. And here you can see two heat maps as examples of cellular activation markers and immune trafficking stimulation. And what you can see on the left heat map is actually in blue that the starting product was mainly blue, which actually was a low expression level compared to the high expression level here for our final product. So this could give you like an overview as like which markers are actually critical for your manufacturing process. And next to that, we will also be able to identify immune trafficking and stimulation markers that were actually not much expressed in the starting product, but were highly expressed in the final product. Zooming in on these genes particular and in total reads, we could actually identify that they were all significantly enhanced. And you can see here that the expression levels of interferon gamma, peripherin, Ramsam B, TNF were actually significantly increased in our final product. But not only these cytotoxicity markers were improved, were significantly highly expressed in our final product, we also were able to identify activation markers such as CD226, as well as TFRC, which is CD71, which were also significantly enhanced. What I also want to highlight on this graph, because I can talk about all these markers specifically, but lag-free and TIM-free and CTL4 were also significantly enhanced. And what I want to actually talk about a little bit here is that while in alpha beta cell therapy, which is commonly evaluated within the cell therapy field, these markers are considered exhaustion markers. There is actually evidence that actually suggests that they are potentially activation markers for gamma delta T cells. So this also demonstrated that our product was actually cytotoxic, but also highly active. And then we could also zoom in on the immune trafficking and stimulation markers such as STAT1 and STAT4, which are essential for T helper 1 and T helper 2 function, as well as TBAT and GATA-free and trafficking markers such as CCR3 and CCR6. Those were all significantly increased within our final product. So we were able to actually, in our final product, we could actually demonstrate that our product was also capable of tissue trafficking as well as immune cell recruitment. So the conclusions that we were able to get from this study using iRepertoire's sequencing platforms we were able to actually demonstrate that our gamma delta T cell products are highly similar from healthy and cancer patients. We were able to produce activated gamma delta T cells from different starting materials, and they preferred an expansion of V gamma 9 and V delta 2 clones. Similar gamma delta TCR profiles were actually observed in a manufacturing product and were not the result of the diversity of the initial apheresis product, were mainly manufacturing driven. The DeltEx DRI products exhibited a highly similar gene expression profile, proving that the process was actually robust and reproducible across multiple donors. And furthermore, we were able to demonstrate increased expression of immune trafficking and stimulation markers in our DeltEx DRI products. So in the end, we were able to demonstrate that our manufacturing process were actually the main driver. So the longer incubation, but also all the our manufacturing process actually were the main driver for the product characteristics, and demonstrated that we were able to produce a robust and reproducible gamma delta T cell products. We are also very interested in what will happen in the end in our patients, and we are also looking forward to utilizing this further, studying this further in patients as well. But overall, we are able to demonstrate with this technology that that the manufacturing process was the main driver. At InnoBio, we are very still excited to eliminate cancer. So please join our mission for Cancer Zero. And that I would like to leave the floor open for questions.
Thank you very much, Mariska, for such a great presentation. And we do have a significant amount of questions, so we can start with the Q&A session. The first question will be, which virus is being used for transduction?
We are using lentivirus for transduction.
Perfect. Tied to that same question, did you check the engineering efficiency throughout the expansion?
Yes, we do check the transduction efficiency and in our final product. Sorry.
Perfect. And is it staying constant throughout the process?
Yes.
Perfect. So we have a second question, which is, do you expect the gamma reads and delta reads to have similar abundance ratios?
What I understand from iRepertoire's technology, and I think iRepertoire can actually explain that in more detail, as that are there and the experts of analyzing the TCR sequencing. But what I understand, there is within this technology a similar abundance of the alpha, beta, and the gamma reads.
Perfect. So there is a question on slide 20. So on the heat map on slide 20, how do you identify those genes? Are those the only genes differentially expressed?
So this is an example of genes that were significantly increased in our final product compared to our starting product. Overall, the data is much more comprehensive and includes about 150 genes. But this is just a selection that we presented here to demonstrate how well you could actually be able to show increased expression of particular genes.
Perfect. So the next question will be, are phosphoantigens, for example, soledronate, used to specifically activate the gamma 9 delta 2 cells?
Our manufacturing process is proprietary, so unfortunately I cannot be able to give an answer to that question.
So another question is, it's great to show there are differences in starting versus product. The person asked, sorry if I missed it, but can you explain what is the product that you're generating?
Our product that we're generating are gamma delta T cells. They're genetically modified, while transducing them with a lentiviral that contains MBNT, which is a DNA repair enzyme.
The other question is, do you often find that healthy donors have a higher starting proportion of gamma delta cells than patients? Or is these cells more activated in healthy donors?
We actually, as demonstrated, we were able to manufacture gamma delta T cells from both donors, and we're successfully be able to produce them and get to sufficient yields in the end. So there's not a particular difference between disease states patients as towards healthy donors in this case.
And we have another question that says, while you report the regulation of activation markers, do you also observe an increase in exhaustion markers?
So we also looked at the exhaustion markers, and I can particularly say is that, well, it's hard, it's still a question which are the particular exhaustion markers for gamma delta T cells. But for example, we didn't see a significant increase of, for example, TIGIT, which is known to be a very late stage marker for exhaustion.
And along those same lines, are any genes significantly downregulated?
I would not say downregulated. It's we're looking at a bulk sequencing. So the genes that are mostly expressed on mRNA level will be presented. So I would say it's hard to say if particular genes are downregulated, but we do see which genes are particularly upregulated in our final product.
Perfect. And in terms of a more expansive view on gamma delta T cells, what other applications or indications could gamma delta T cell therapy be used for?
So in this case, we're showing how it could be used for GBM in cancer patients. We could also be able to study this further in other solid tumors such as breast or prostate. This DeltEx DRI would be also an cell therapy for those indications, for example. We are currently also we also have another trial in which we're mainly focused on, and that's our IV100 trial, where we're actually looking at highly risk relapse leukemia patients, and where we actually treat with an unmodified gamma delta T cell product to improve progression-free survival in leukemia.
Perfect. So we have another question that says, could you see any effects on the functionality if different clones were expanded per different donors?
No, we couldn't particularly highlight if there was a specific. Maybe the data set is still too limited. We need to analyze more. But I do think that it gave us a good indication on the markers which we should be looking at and focusing on. Although I do see that it's going to be a large set. There are a lot of important markers within cell therapy to look at. And as what I said, for potency, it's important not just to only focus on a killing assay, but we also need to really focus on more expression levels of our products and be able to demonstrate the definite mechanism of action.
Perfect. So along the same lines, in the final product, is more enriched for non-gamma 9 delta 2 T cells. Would you expect they will perform better in vivo for tumor killing compared to gamma 9 delta 2 enriched product?
We do expect that V gamma 9 and V delta 2 gamma delta T cells would have improved killing in cancer. But I still think that the V gamma 1 is still essential as well, and it's just not one fits all in the end. but we do, you know, there are articles out there that's mainly V gamma 9s and V delta 2s are highly cytotoxic. So that might indicate to be better.
Exactly. So, for example, do you consider a specific set of markers essential? If so, which ones will they be? And if you have tried to tracking those clones longitudinally across the different patients?
So that's an unknown question because we focused on build sequencing, so we cannot really single out these clones. We can only look at the overall expression and give our best guess. But I didn't see a particular upregulation or downregulation compared to products that contain a little bit more V gamma 1, V delta 2, sorry. I'm mixing up them all. Sorry, my apologies. What I actually meant was I didn't see a difference for a delta 2 product.
Perfect.
One.
So we have a final question, which is, how do you plan to utilize all of this data in your product pipeline? So for example, will it inform potency or other lot release specifications?
So how I see this is that it's very important to further characterize your cells. And if you are in the cell therapy field, you really have to figure out what the mechanism of action is of your product and which, you know, genes and which potency markers are actually essential. You don't want to go in, you know, what the actually happened with alpha beta, where they were focusing only on interferon gamma, and then end up, it's going to be probably going to be an overview of multiple markers in the end, and you need to start tracking them and understand more about and cell therapy. And I think that's the most important thing, is that cell therapy, it's so difficult to eventually figure out what's the mechanism of action is. And which ones are important, but it's key to keep them all in, you know, keep track of them all as much as possible.
Exactly. So yeah, that sounds super interesting. And we wanted to thank you again, Mariska, for such a great presentation and answering all of these questions. If we didn't get to your questions or you have any additional one, we will reach out to you by email. And if you have any other questions about what we presented today about iRepertoire's products or services, please visit iRepertoire.com or reach out directly at info@iRepertoire.com. And with that, thank you very much and have a great rest of your day. Thank you, Mariska, again.
Thank you. Thank you so much.